Welcome to the NPTEL Immunology. Okay. I hope my video is visible as well as I'm audible. So, hello everyone. Welcome to the NPTEL Immunology live sessions. So, I'll be sharing the screen and we'll get started. So, so I am basically I am Seemanti Aditya, a Ph.D. scholar in the Department of Biotechnology at Indian Institute of Technology, Madras. I'll be your teaching assistant for taking live classes with respect to any doubts that you will be having with all the video lectures that you are getting on the NPTEL platform. So I'll be taking live classes every Friday from 7 p.m. to 8 p.m. So today we'll be discussing certain concepts that is related to week 1 content. Also, I'll be discussing some questions that would be helpful for you to answer maybe the assignment that you're getting. So before starting, I'll just share a poll just to know that what kind of degree you all are pursuing, so that I can make the content accordingly. So I'll be launching a poll. So please answer the what kind of degree you are pursuing. Okay, so I see a lot of PhD scholars taking and as well as BSc. So yeah. Okay. So let's begin with the course. So I hope you have watched the week 1 contents and like any doubts that you will have, you can discuss over here. So before that, maybe I'll just go through certain concepts related to week 1 lectures that is already published. So let's begin. So the course is taken by Professor Agnayo Ganguly and Professor Sudeep Kumar Ghosh from the Department of Biotechnology at Indian Institute of Technology, Kharagpur. Today, I'll be discussing about a brief introduction to immunology. So what is immune responses? Basically, any response to foreign particles, the body's response to foreign particles is known as an immune response. Now, this immune response can be because of an integrated network of cells, molecules, and organs which are evolved to, protect our cells or multicellular organisms from pathogens. So this kind of immune responses confer a state of protection, which we call as immunity. So this immunity, the word immunity comes from a Latin word known as immunis, and it is provide like different people. We have seen that so some people are genetically blessed with a better immune system, and some people they have less strong immune system. So this is also again to some extent dependent on what genes you have inherited from your parents. So the basic concept of immunology came from Edward Jenner. So he is known as the father of immunology. So he did was he inoculated fluids from cowpox pustules. So he found that people were dying from smallpox. So he saw that, okay, so if we are isolating some pustules and allowing the people to get exposed by those pustules by a process known as variolation. So variolation includes letting the people smell those kind of pustule swabs or this infected wound swabs from cowpox virus. So people who were exposed to those kind of pustules, people who were exposed to this cowpox virus associated disease tissues, he found that those people were protected from smallpox as well. So from here started the origin of immunology, or the study of the immune system. So what kind of protection did the body produce in order to protect people from getting smallpox infection? So here, so carrying forward this studies, Louis Pasteur, he demonstrated that if you are administrating any kind of attenuated strain, so attenuated means any weakened strains of any kind of pathogen, bacteria or virus, it might confer an immunity against future infections and disease. So he called the attenuated strain as a vaccine in honor of Jenner's work. So Pasteur, we know that Pasteur had developed certain vaccines, including rabies vaccine and cholera vaccine. So he put forward the work of Jenner, and he inoculated. Here you can see a picture of Joseph Mister. He was the first person who received the vaccine for rabies. So he was basically bit by a rabid dog multiple times. And rabies vaccine, like most of the vaccines, work on the principle that it will prevent the disease from occurring. So vaccines are usually taken before you have encountered any disease. But rabies vaccine is one kind of exception that can be taken after one kind of exception, or few of the exception vaccines that can be taken after after you have been bit by the rabid dog. So Joseph Mister was repeatedly bit by a rabid dog, and so here is Louis Pasteur, who is watching him getting the shot for the rabies vaccine, and basically he lived. So he grew up to take care of the Pasteur Institute later on, Joseph Mister. So from here came the concept that, okay, so if we are administrating any kind of weakened strains or dead viruses, so body can still recognize those those small particles when, if they are further in the future lifespan, if that particular virus is getting infecting the person. So humans have that kind of memory to recognize future infections and protect from the disease if we are already vaccinated. So the idea of vaccine is very important in terms of the scenario that we are facing, or we have faced in the past couple of years. So we all have heard the term herd immunity. So what is herd immunity? Now, any kind of like vaccines, when we are administrating to a large population, so it is not possible that in a very quick period of time you can vaccinate almost all of the population. But herd immunity works on the concept that if enough people are vaccinated, so if most of the people or enough people are vaccinated, it can also protect the individuals and communities who are susceptible to the disease through herd immunity. So we can see from the picture that when people are not vaccinated, so blue colored cartoons represent susceptible community, whereas the red colored cartoon shows infected community. So when people are not vaccinated, so they will be more prone to come in contact with disease susceptible people will be more prone to come in contact with already infected people. So this will lead to further spread of infections. But if we are vaccinating people, most of the people are vaccinated, then we can see that there is a buffering system. So there is no direct relation between the disease susceptible person and the infected person. So there is a person standing in between the disease susceptible and the infected person. So this actually reduces the chances of infection to a great level. So this concept was utilized for this COVID vaccination, where enough people, like most of the people, were vaccinated within very short period of time, and that protected basically the disease susceptible people from getting the infection from already infected individuals. So this is another very interesting concept for immune responses and immunity. Now, so what are there are certain small terms that are associated with immunity, which I would be willing to discuss. So what is an antigen? So antigen is a foreign particle that is capable of binding to an antibody. And so there is another term called immunogen. So immunogen is a particle that is able to elicit an immune response, but it might not be able to bind to an antibody. So but antigen is a foreign particle which is capable of binding to an antibody. So basically, the sequence of antigen will be very complementary. Some certain sequences of antigen are complementary to antibody, and it will allow the interaction between antigen and antibody. Whereas antibody is a protein molecule that is produced inside our body. So antigen is a foreign particle, and antibody is a molecule which is produced inside our body in response to the antigen or the foreign pathogen, and it will allow the binding and neutralization of antigens. So basically, antigen antibody interaction is essential for eliminating the pathogens and neutralizing the antigens, and basically not allowing the disease to take place. So what is an epitope? So epitope is a region of an antigen. Now we can see in the diagram. So this is an entire antigen. Okay. So we have like an antigen is a big molecule, not a big molecule, but yeah, compared to what is associated with immune responses. So antigen will have a lot of structures, but only a certain region of antigen is actually recognized by the antibody. So okay, here it is. So this, okay. So epitope is a region of antibody that is antigen that is going to be recognized by the antibody. Now here I have made a little mistake. So this is actually paratope. Okay. So the region of antigen that is recognized by the antibody is known as epitope, whereas the region of antibody that interacts with antigen is known as an paratope, which I have mentioned as immunogen. This is actually paratope. I'm sorry. And immunogen is actually a molecule that is responsible for eliciting an immune response, whereas antigen is a foreign particle which will bind to the product of an immune response. So product of an immune response is basically an antibody. So yeah, epitope and paratopes are regions of antigen and antibody respectively that is going to interact with each other. And this interaction is essential for better selection of this like better specificity of antibody towards the antigen. Is anybody saying anything? Okay. So better selection of the antigen and basically neutralizing the antigen. So the components of immune system includes innate immunity and adaptive immunity. So we have all studied about this. Is very basics of immunology that I am dealing with. So innate immunity is any immunity that is non-specific and shows an immediate response to any kind of evader. So basically, if you are getting a cold, if you're getting a wound, if you're getting hurt, or if you are like exposed to any kind of, we are always exposed to so much of pathogens, so much of viruses and bacteria all around us. So our innate immunity is always maintained at basal levels. So we will have an immediate response to any kind of evader inside our body. So those immediate responses basically first provided by the anatomic and chemical barriers of our body that includes skin, tears, then the mucous membranes, etc. So this provides the first line of defense. And innate immunity is basically non-specific immunity. So it is not going to be specific to any kind of pathogen. So all viruses or all bacteria will be treated equally. So equal kind of responses will be there. So there is no specific antibody which is going to be generated against any specific virus in case of innate immunity. So innate immunity is conferred by macrophages, dendritic cells, NK cells, neutrophils, eosinophils, basophils, etc. So macrophages, dendritic cells, NK cells, they are responsible for phagocytosis, associated with engulfing the content or engulfing the pathogens, and then releasing certain inflammation markers like cytokines and chemokines. So these are known as mediators. Cytokines and chemokines, these are known as mediators because they help, they are produced by the sensor cells. Sensor cells include macrophages, dendritic cells, NK cells, etc. So whatever cells that are recognizing the pathogen, so those cells are known as sensor cells, and they will synthesize this inflammation markers like cytokines and chemokines. So these are known as mediators. So this will allow the immune response to proceed further, like it will intensify the immune response. And in contrast to this innate immunity, which is non-specific and is associated with phagocytosis and inflammation, adaptive immunity will take some time to develop. So as soon as a pathogen enters the body, it is not developed immediately. So we don't see, like, if you are attacked by a very serious pathogen, suppose if you are attacked, suppose if viruses like COVID or viruses like influenza attacks you, it's not going to start immediately. It will take some time to develop, in contrast to the innate immunity, which is starting very immediately. But it will provide specific immunity to those specific pathogens. Now, this is very important, that adaptive immunity, although it takes some time to develop, so after it has developed, it will provide the specific immunity. So what happens, like if you are providing some specific immunity, so later on, if this pathogen enters the body in some other time of your life, so this specificity will again be recognized, and the immune response will act as a much more stronger way to eliminate that pathogen. So we say that if you are getting chicken pox once in your life, maybe you will not get it next in anytime sooner in your life. So again, so this is because your adaptive immunity produces a specific immune response against those pathogens which are remembered by your system, and those memory T cells and memory B cells help in fighting the pathogen later on in your life. Now, adaptive immunity is conferred by two kinds of cells, basically the B cells. This is known as the humoral immunity, because B cells are producing antibodies. So what we know as an antibody, what I discussed, that is a molecule that binds to antigen. So antibody is produced by B cells. And humoral, the term humoral comes from the word humor, which means blood. So antibodies are secreted in blood. So so any immunity, adaptive immunity, which is conferred by B cells due to the synthesis of antibodies, is known as humoral immunity. Whereas immunity that is conferred by T cells is known as cell-mediated immunity. Now, adaptive immunity can also be conferred by dendritic cells. So you see, dendritic cells has a position here in innate immunity as well as in adaptive immunity. So dendritic cells basically bridges the gap between innate immunity and adaptive immunity. We will see how. And it is also an antigen presenting cell that will basically go and engulf all the pathogen from in this innate immune response. It will go and engulf the pathogen, show phagocytosis, and then it will process the pathogen and show it to the B cells, or show it to the T cells. So it is going to bridge the gap between innate and adaptive immunity. Now, adaptive immunity is associated with the release of antibodies, which is by plasma B cells. So there could be two kinds of B cells. One is membrane-bound B cells and plasma B cells. So plasma cells, they are synthesizing the antibody in the blood, whereas in membrane-bound B cells, we will have the antibody which is attached on the membrane of the B cell. So initially, when the antigen is recognized, so B cell will recognize the antigen on the basis of the antibody present on the membrane. Later on, after it has recognized the antigen, after it has undergone clonal selection to finally become the subtype that is specific to that antigen, it will convert to plasma cells, and this will result in the synthesis or release of antibodies from its membrane. So antibodies will be released in the blood and will go and neutralize any antigen that it finds. And also adaptive immunity is associated with cytotoxicity. So this is conferred by T cells. Again, T cells, we have seen that there are two types, T helper cells and TC cells. So TC cells are cytotoxic T cells, and these are associated with killing the cells. So innate immunity and adaptive immunity are like both are important for our immune system to work properly, because these are interrelated and correlated. If if the pathogen is not so harmful, it will be tackled by the innate immunity. But if it is not being tackled by the innate immunity, then the adaptive immunity comes to play. And adaptive immunity can come to play only because innate immunity will be functioning properly, because adaptive immunity cannot function independently of the innate immunity. So also, we had seen that in the lecture, we could see that all these vertebrate immune cells that we are seeing, these macrophages, dendritic cells, NK cells, B cells, T cells, these are all the components of immune system. And these cells are the immune cells, which are, all cells in our blood is basically developed from pluripotent hematopoietic stem cells. So any kind of stem cells can give rise to, we know that it can give rise to different, it can differentiate into cell types that are having specific characteristics. So all the kind of cells that are developed, that are present in the blood, comes from hematopoietic stem cells, and this can be divided into two broad categories, namely the common lymphoid progenitor cells and the common myeloid progenitor cells. So lymphoid progenitor cells are responsible for secretion of B cell, T cell, NK cells. NK cells are, the full form is natural killer cells. These are also responsible for identifying the pathogen and killing it by killing the pathogen. And ILC is the innate lymphocyte. So this particular common lymphoid progenitor cells, B cell, T cells, NK cells, etc., are mainly associated with the adaptive immune response. B cells and T cells are associated with adaptive immune response, although NK cells and ILCs to some extent also responsible for the innate immune response. And common myeloid progenitor cells can again be divided into two categories, like granulocyte or macrophage progenitor cells and megakaryocyte or erythrocyte progenitor cells. So granulocyte or macrophage progenitor cells will be responsible for the origin of neutrophils, eosinophils, and basophils, and whereas megakaryocyte or erythrocyte is responsible for the development of erythrocytes. Apart from that, the immature dendritic cells are also coming from the myeloid progenitor, which again will get developed to mature dendritic cells. So both these progenitor cells are responsible for responsible for the development of the immune cells in our body, both the innate immune cells and the adaptive immune cells. And this kind of there could be as we saw, that dendritic cells may be an intermediate mediator between the innate immunity and the adaptive immunity. So how could the innate immune response how the innate immune responses are originating in an infection, we can see in different kind of pathways. So pathogen, whenever it enters our body, so it has certain molecules expressed on its surface, which are called as PAMs, or pathogen-associated molecular patterns. Now, pathogen-associated molecular patterns, this is recognized by certain receptors or certain protein molecules expressed on the surface of the phagocytic cells. That could be macrophages, dendritic cells, or neutrophils. So PAMs are recognized by PRR. PRRs are again receptor molecules present on the surface of these phagocytes. So PAM and PRR interaction is essential for the phagocytosis of the pathogen. So then what will happen is the macrophages or the dendritic cells will engulf the pathogen, and it will result in the killing of the pathogen by degradation in the lysosomes. So lysosomes are again acidic vesicles present. These are membrane-bound vesicles having acidic hydrolytic enzymes. So once it is engulfing the pathogen, it will lyse the pathogen by fusing it with the lysosomes, and this hydrolytic enzymes will cause the killing of the pathogen. So apart from that, pathogens can also be eradicated due to the synthesis of antimicrobial peptides. So these antimicrobial peptides are again synthesized in the blood due to phagocytosis or due to this innate immune responses, and this will result in the killing of pathogen by damaging to the membrane. There will be pores in the membrane, and finally the bacteria will be so the pathogen will be killed. Apart from that, we also have certain molecules that are recognizing this PAMs. So these are these are surfactant protein A, surfactant protein B, mannose binding lectin, C-reactive protein, and complement proteins. These all are proteins. These are known as opsonins. So what are opsonins? So opsonins are any molecules that are soluble and that will enhance the phagocytosis of the pathogen by binding to it. So these molecules are normally present in the blood. So when a pathogen enters, it will recognize the pathogen-associated molecular patterns or PAMs. So these molecules, opsonins, will recognize PAMs and will bind to the pathogen. And once it is bind to the pathogen, it is known as an opsonized pathogen. So this pathogen, which is coated with all these proteins on its surface, will be easily recognized by phagocytes through its PRRs, because it is also having the PAMs that are associated with the pathogen. So opsonization will enable the phagocytic cells to better recognize the pathogen and engulf the pathogen and cause phagocytosis. So these molecules are known as opsonins because it is helping the phagocytic cells to undergo phagocytosis. So phagocytosis will take place, and ultimately the pathogen will be killed. Then we also see that this phagocytes activate to produce antimicrobial compounds, which is followed by the secretion of inflammation promoting cytokines and chemokines. So any kind of after phagocytosis, they will produce antimicrobial compounds that will kill the pathogens and also produce cytokines and chemokines. So these are mediators. This will allow other phagocytic cells to come to that location and will help in intensify the immune responses. So as we also, there is another important component of innate immune system, that is the complement immune system, which we will study in the later on classes in details. So for now, it is we should know that complement will destroy the membrane. So what happens is there are several complement proteins. So it will recognize, it will go and bind to this pathogen, and all these complement proteins forms an oligomer like structure, and it will form a pore on the surface of the bacteria that will result in the lysis of the bacteria, and the cell will die. So complement pathway is also activated during immune response, during innate immune response, and is essential for providing protection against different pathogens. Now, how dendritic cells are actually mediating the response to why is it known as the bridge between innate and adaptive immune response? So dendritic cells are also basically can act as a phagocytic cell, and it can engulf the pathogen. So it will have PRRs on its surface that will recognize the PAMs on the surface of the pathogen. So it will activate the phagocytosis and the signaling pathway. So once once the phagocytosis takes place, so the pathogen will then get degraded, or maybe it is still intact. So after after it has undergone phagocytosis, then the dendritic cell will migrate to the lymph node carrying the intact or degraded pathogens. After it has migrated to the lymph node, the antigen fragments that is bound to the cell surface. Now it also has MHC molecules on its surface. So every immune cell has an MHC molecule. So it will bind to the antigen fragments will bind to the antigen fragments will bind to the MHC proteins which are expressed on the surface of the dendritic cells, and that processed antigen, after interacting with the MHC molecule, will be recognized by the T cell receptor, and the T cell receptor will finally bind to this process antigen and cause the adaptive immune response and the killing of cells. So you can see that the dendritic cell will show the phagocytic activity as well, and also act as an antigen presenting cells. So there are three different kinds of antigen presenting cells: macrophages, dendritic cells, as well as the B cells. So all of this have this kind of property where they are, but only macrophages and dendritic cells can phagocytose. B cells do not have the property of phagocytosis. So they will they will undergo phagocytosis and ultimately show the antigen to the T cells. So antigen after recognized by the T cell will result in the killing of killing of the cells. So cytotoxicity will be followed in the next steps. Now maybe I'll just give a poll of one question. So this is related to something that I just discussed. These are very simple questions. I think you'll be able to answer. So after that, maybe I'll just I have just some few more slides. So are you guys there? So yeah. Okay, Suparna. Thank you. Somebody had raised a hand. So did you understand? Did you have any doubt till now? Yeah, I had gone through certain questions that was, asked in the forum. So I was trying to answer those questions with this slides. I hope like some of the questions that were asked in the forum is answered. If not, you can still ask me some questions.
Excuse me, ma'am.
Yeah, hi,
Ma'am, I just have a small doubt, ma'am. Ma'am, you just told about variolation in the first slide, right, ma'am? About
yes.
So, is it an innate immune response? Will it activate an innate immune response or an adaptive immune response, ma'am?
It will activate actually an innate immune response initially,
Okay.
and because we are actually in variolation, what happens? No, like what he did is he used cowpox pustule to treat smallpox. So innate immune response is initially activated. Later on, there will be certain antibodies that will be synthesized. Adaptive immune response will also be activated, but the antibody will not. Sometimes what happens is antibody can be cross-reactive. So, what he did is he used cowpox pustule to treat smallpox. So there will be certain regions of the antigen that will be similar in both kind of viruses. So after the innate immune response has started, when the antibody against cowpox is generated, that antibody will have certain regions that will bind to the antigen of the smallpox. So it will show kind of not like entire specific response, but yeah, there is this term called cross-reactive antibodies. So this is one of the examples that where cross-reactivity comes to play. Yeah.
So it's both innate and active, right,
Yes.
ma'am?
Hmm.
Another doubt, ma'am.
Yeah.
Ma'am, you told about opsonins, right, ma'am?
Yeah. Yeah. Yeah.
Opsonins, like, do they help in attracting the macrophages towards the pathogen? Or
Yeah,
they,
yeah. That is the role. Like opsonin is basically binding to the pathogen and will help the pathogen to be, to be recognized by the macrophages more efficiently. Like otherwise, macrophages will recognize the pathogen if it is not bound by opsonins, but opsonins they are enhancing the capacity of macrophages to recognize. So it will definitely attract. I'll see the poll. I think all of you have answered correctly, right? Okay, so some of you have answered like which cells act as a bridge between innate and adaptive immunity as B cells. Why? Like, why do you think that B cells will be acting as a bridge? B cells are actually synthesizing and specific antibody, right? So it is not going to undergo, like it is not going to go through the phagocytic pathway. It is not going to show a non-specific immune response anytime. Like innate immunity is associated with non-specific immune response. It is associated with phagocytosis. So B cells are not, B cells are not going to uh, do that. It is going to be very specific. So it's going to be dendritic cells, okay? Because dendritic cells can act as both phagocytes as well as and adaptive immune response. Like it when it will present the antigen to the T cells.
ma'am, but you also told that B cells can also act as presenting cells, ma'am, antigen
Yeah, yeah,
presenting.
antigen presenting cells, but antigen presenting cells could be dendritic cells, macrophages, as well as B cells. Okay, but B cells will not undergo like it is not going to cleave the pathogens into small like small peptides, and then like it is not going to be non-specific. B cell is not going to show any non-specific immune response. It will just like it will present the antigen to the T cells, and on the basis of what macrophages or dendritic cells has presented to the B cell already. Like B cell will come later on after macrophages and dendritic cells have come into play. That is what I have the knowledge. Like B cells, they are not going to show any kind of non-specific immune responses. It will show it will present the antigens to the T cells only after it has undergone the innate immune responses from macrophages and the dendritic cells.
Excuse me. Hello, madam.
Yeah, hi.
There's one my question is that MHC complex is required for T cell activity. Okay.
Yes.
Now this antigen, suppose antigen presenting cell, they are presenting part of the antigen to the T cell. Okay. Now in the latter case, whether MHC plays any role, is it exclusive that T cell requires MHC complex for its
Yes.
binding?
Yes. Yes.
Then how come antigen presenting cells providing some information to the T cells? Because in that case, MHC is absent.
No. So antigen presenting cell is presenting. The term antigen presentation means that antigen is processed and presented with respect to MHC like after getting bound to the MHC molecule.
Or
Any
then
antigen presenting cell will
antigen presenting cell, they are providing the information to the MHC. Then
antigen No, antigen presenting cell, what it is doing is, so it will have, one second, I'll just share this. So see, antigen presenting, here I'll start. So see, you are seeing this dendritic cell, right?
Yeah.
This dendritic cell will engulf the pathogen
Okay.
inside it, and pathogen will be cleaved into small, small pieces.
Hmm.
Then it will have an MHC molecule
Hmm.
that will bind to this small peptides that is already
Okay.
cleaved.
Okay.
Okay.
Understood. Understood.
Yes,
Understood.
and then
Okay.
it will bind to the T
Okay.
cell
T cell.
receptor.
Okay. Many thanks. Many thanks.
Yeah.
Can you just explain that thing again? I don't understand the role of dendritic cell in.
What?
I didn't understand that. Can you just explain it again? What you just that antigen presenting, like the bridge,
Why it is
dendritic cell.
called the bridge, right? So, okay, let me erase it. So we have, suppose we have a dendritic cell over here, and we have a pathogen over here. Now, all these cells, what they do is, this innate immune cells like dendritic cells or macrophages or NK cells, they have the property that they will engulf the pathogen. Okay. So they are engulfing the pathogen. Now, once they have engulfed the pathogen will be processed into small, small peptides or proteins, like whatever is the pathogen inside the cells of the inside the dendritic cells. Now, this engulfing property is very exclusive to some kind of cells. These are only just neutrophils, dendritic cells, and macrophages. They have these properties for engulfing the pathogen. Now, after the pathogen has been engulfed, then what it does is dendritic cell will have certain molecules on its surface, which is known as MHC. So this is major histocompatibility complex, and these molecules will then bind to the small peptide fragments, okay, that is already cleaved after phagocytosis. So these small peptide fragments then will be, so this is called processing. So whenever a pathogen is cleaved into smaller fragments, so these are known as processing of the antigen. Now, after processing, these peptides are, expressed on the surface of the dendritic cell through MHC, and this will be recognized by the T cell receptors on the surface of the T cells. So now T cells belong to adaptive immunity, right? And this is the this is the, okay, again I'll draw. This is the peptide. Dendritic cell will bind to the T cell through the T cell receptor. Now this T cell belongs to adaptive immune response. This belongs to innate immune response. So you see, the dendritic cell is basically presenting it to the adaptive immune response, and it is going to act as a bridge. Now what B cell will do is, B cell will have an antibody on its surface. Okay, it is not going to show any phagocytic activity like the dendritic cells or the macrophages. So B cells will recognize antigen. It can recognize antigen as a whole. So it does not require antigen processing. So it will recognize antigen as a whole, and then after recognizing the antigen, this B cell receptors or this antibody like molecule, this will then go and present this antigen to the T cell receptors present in the adaptive immune cell. So B cell receptor basically belongs to adaptive immune cell as only. So it is not going to, it will go and present it to T cell receptor. So T cell receptor is also again an adaptive immune cell. So it is not going to act as a bridge. Bridge will be only found by the dendritic cells. Okay.
Yes, ma'am. But I didn't understand, ma'am. The dendritic cells is already presenting it to the T cells. Then why is the B cell again presenting the thing to the same T cell, ma'am?
So it is like, why so many, then why would we require so many cells? Why macrophages are also doing the similar kind of thing? Why neutrophils are also doing the similar kind of thing? Just to increase the immune response, just to
So
increase.
it is a backup plan?
It's not a backup. It is like all these cells, they require each other. So it is not like one cell will not work, so the other cell has to work. That it is not like that. It is all the cells has to work in a unified fashion, so that the entire immune system works properly. So if each of it has a specific, each of it needs to work as a team. So macrophages will also go and phagocytose the cells. The B cells will also go. So more number of cells that are there, more number of stronger will be the immune response, and more faster will be the immune response. As I know,
So the B cells are not breaking down the antigen, whereas dendritic cells are breaking down the antigen and forming the MHC complex to get activated, and then the next process start. Am I right?
Yes, that is, yeah.
Doing it just to increase the efficiency of the response to
Yes.
the antigen as soon as possible from the body, right?
Yes, like to clear the infection, because if there is just one cell, then you will know so much of it will require so much of time to clear out the infection. But if there are many cells working together as a team, so it will be cleared out much more sooner. So yeah, so all the cells need to function properly and carry out this activity. So all these cells require a team kind of action.
One more doubt I have, that PAMPs that is present on the pathogen, which is recognized by the dendritic cell and macrophages, are the same PAMPs are also helping the B cells to recognize them, or is it something else which is initiating the B cell to identify the antigen?
PAMPs are actually B cells that I have to once check whether B cell also recognize PAMPs, because as far as I know that it is only recognized by PRRs from the this macrophages and dendritic cells. So
that means the B cells are identifying the antigen only by the antibody present on their surface.
Yes.
Not because of the special type of protein present on the pathogen. That protein is only meant for these phagocytic cells, if as per your...
I think so. Like not this specific kind of protein like PAMs, because any kind of protein that is like not our protein, so B cells can recognize those proteins, but not I'm not sure that whether it recognizes specifically PAM or any like changes in the other protein molecules. So all these antigen molecules or all this pathogen will have different composition of the proteins on their surface. Like so bacteria has this lipid by LPS layer, or any kind of layer that is not present inside our human system normally. So whether it is recognizing just the PAMs or like all these kind of LPS or everything, like I am not sure about that. But I'll need to check. I'll just maybe in the next class I'll answer that.
Okay, no problem. Thank you.
Okay. So if anything I'm saying wrong, you can also add on and say that okay, maybe you have some idea about this, any other person in this class. So if you know about something that I don't know, maybe you can like share with us that thing. Basically we are all learning here, so yeah.
After the dendritic cells are migrated to lymph nodes,
Yeah.
how it will excrete?
After the dendritic cell is migrated to the lymph nodes,
Yeah,
how it will get excreted?
yeah.
Why do you want, like, it will be like, these cells are not having much lifespan, so it will, after doing its job, most probably it will die. Why it will get excreted? So okay, these cells are mostly associated with, small lifespans, all these WBCs and lymphocytes. So once it is doing its job, it will it will die basically. After it is died, then it will be excreted, I think, through the uh,
So the spleen, ma'am. Spleen
through
and
the
then liver.
okay,
Graveyard.
okay,
Graveyard of the cells.
yeah, so yeah, it will die basically. So okay, so we will go forward to what is like how the adaptive immune response is mediated. So we have foreign proteins or infectious agents. If it is exposed, if any kind of animal is exposed to this foreign proteins or infectious agents, there can be two kinds of adaptive immune responses. It will be humoral response or cell-mediated response. So in case of humoral response, we see that B cells are coming into action. In case of cell-mediated response, T lymphocytes are carrying out the function. So what happens is, so B cells, you see, these are B cell receptors or antibody, like we also known as antibody, like there could be antibodies on the surface of B cells. So these are recognizing antigen as a whole. And after it is recognizing the antigen, this B cell gets matured enough and forms antibodies that are secreted. So this is known as membrane-bound B cells, and these are plasma cells. So plasma cells secrete the antibody in the blood, and it will go and bind to the other antigens which are already circulating, and will result in antigen elimination. Whereas in case of cell-mediated immune response, T cells will recognize antigens which are presented by MHC. So this antigen, after being presented by the MHC molecules, will result in the activation of T cells. And T cells is also responsible, if T cells are getting activated, it is also responsible for cytokine secretion. So cytokine and chemokines, these are all mediators. And this are responsible for enhancing or strengthening the immune responses by allowing the B cell to get matured to plasma B cell, and also recruiting the macrophages and dendritic cells to that surface, to that basically to that point. And so after the T cell receptors are getting activated, it can bind to this antigen and also show the killing of the infected cells through apoptosis or cell death. So these adaptive immune responses and innate adaptive immune responses can, like, this by blood, like through blood, it is synthesizing antibody known as the humoral immune response. And cell mediated is like when T cells are recognizing the recognizing the antigen processed by the antigen presenting cell and presented by MHC molecule. So it is directly causing the cytotoxicity of the cell. It's not getting, anything is not getting released into the blood. So it's cell mediated response. So on the basis of that, I'll also have some other questions maybe. So yeah, so the question is this, I had discussed right now. Okay, so maximum of you have given the correct answer. So the common lymphoid progenitor cells, as I discussed, like it is going to give rise to B cell, T cell, and NK cells, whereas myeloid progenitor cells will give rise to all these immature dendritic cells or erythrocytes, neutrophils, basophils, or eosinophils. So yeah, that was correct. Uh, so to the next question, I'll just launch quickly. Actually, I had seen that somebody had posted in the forum about what is mediators. So you see in the question there is like inflammatory mediators are proteins that are secreted by the sensor cells to amplify the immune response. So all this basically it is going to recruit other antigen other immune cells to the site of infection and increase the immune response.
Excuse me, ma'am.
Yes.
Yes, ma'am. Actually, you told that T cells are cell-mediated, induce cell-mediated response, ma'am.
Yes.
So that means that these antibodies, so when the MHC, when the dendritic cell, they present the peptides, the MHC class,
Mm-hmm.
MHC molecules, they
Mm-hmm.
get activated and then they remain in the cell and eliminate the antigen or what, ma'am?
So you're asking that whether the peptides are remaining inside the like in the cell only in the dendritic cell. Is that
No,
what?
I'm talking about the T cells, ma'am. Like how are they cell mediated? I didn't understand. Like how is it cell mediated response?
So okay. So basically, B cells what they do is they are secreting the antibodies. No, in the blood. They are not going to directly. First, it is going to bind to the antigen. The antibody that is present on the surface of B cell, it will bind to the antigen. Okay, someone is sharing screen, and then it is going to. So yeah, so once the B cell is bound to the antigen, it will get activated, and then it will release antibodies in the blood. So that antibody is going to neutralize the antigen. But in case of cytotoxic immune response or cell-mediated immune response, T cells is directly interacting with those peptides which are expressed by MHC on the surface of antigen-presenting cells, and it is causing the death of that cell. So it is causing the death not by secreting any molecules. It is directly by the interaction of the T cell receptor with the, these processed antigens. So that is why it is called cell mediated.
So it will release cytokines and chemokines through that.
Yes, no, it is going, see, again, immunology is not like, okay, so it is just having just one function, particular function that, okay, I will just kill the cells and not do anything else. It will also help in the, as I said, that it is going to, the entire immune system needs to work as a team. So the T cells is also going to activate the B cells so that both the cytotoxic immune response as well as the cell mediated immune response as well as the humoral immune response comes into play together at the same time. So it will be like better elimination of the pathogen. So it will also release cytokines. So we are also seeing that macrophages are phagocyting the cell and also releasing cytokines. B cells are also, interacting with the antigens and getting activated to form plasma B cells and releasing antibodies. So T cells are also going to, act with the B cells and the T cells are also going to act with this antigen presenting cells, kill those kill those antigen presenting cells who are presenting the peptides, and also release cytokines. So all of this will work together. So cytokines, chemokines, everything, these mediators need to be released for proper amplification of immune response, for proper recruitment of the immune cells to the site of infection.
So you mean to say that all that macrophages, T cells, all of them secrete these molecules to increase the.
Increase the immune responses. Yes, there are mediators which are, yeah.
Okay, okay, ma'am. Thank you.
Yeah, thank you. Okay, so one more question. I'll just quickly launch it. So yeah, Most of you have given correct answer. Some of you have written just cytokines. No, both cytokines and chemokines are inflammatory mediators. This is going to act as a act for recruitment of different. Yeah, somebody has a question. Okay. So the last question the second last question that I'll just launch. Most of you have answered characteristics of inflammation as migration of leukocytes out of the circulation. No. So why not? Okay. So let me again share my screen for a quick period of time. Somebody has raised a hand. Okay. Again. So what are the characteristics of inflammation? Do you know? The inflammation is associated with redness, swelling, heat, and redness, swelling, heat, and what was the last one? Redness, swelling, heat. One second, let me check. Redness. So another one was there. I'm not able to recall. So these are the some characteristics of inflammation. So swelling is actually associated when leukocytes are migrating to the site of infection from the circulation. So leukocytes are in the circulating blood. Okay, and suppose here is your infection, and so there is more entry of leukocytes or neutrophils to this surface of infection. So here it is going to be swelling will be shown. So so migration of leukocytes is not uh, a is actually a characteristic of inflammation. So characteristics of inflammation includes migration of leukocytes out of the circulation. Pain, basically that is pain that is associated with inflammation. Okay, and the correct answer for this is secretion of antibodies, because inflammation is associated with innate immune responses. Secretion of antibodies is associated with adaptive immune responses. So antibodies are very specific. It is not going to be synthesized initially in the initial stages of infection. So all this pain and swelling, cytokine production, migration of leukocytes, this is going to generate these 4 categories of inflammation that results in redness, pain, swelling, and heat. So whenever you're getting a mosquito bite, whenever you're getting like there is a redness there is little swelling there, you will also, if you touch it, it will become warm. So these are all characteristics of inflammation. And migration of leukocytes to that point, like outside of its circulation to that site of infection, will result in swelling, because it will result in the accumulation of leukocytes and more number of cells in that area. So that is resulting in the swelling. So this is not the correct answer. The answer is secretion of antibodies. Now maybe, so I am almost done with the class. So
Excuse me, madam.
Yes.
Regarding the last answer of the question, secretion of antibodies. Okay. So if there is a reinfection occurs, say for example, some memory cell causes a particular response for that particular protein. So there will be some kind of inflammatory responses, pain, swelling, whatever it is, maybe some temperature rises. But in that case, the only associated functional body is antibody.
Yeah, but there will also, you see, you get, whenever there is a secondary infection, you still get fever, you still have some kind of inflammatory responses. So the primary acting cells will be the adaptive, like it also has to be in a particular fashion. So as soon as a pathogen will enter your body, okay, your B cell, memory B cell is not going to totally go and like understand that, okay, this is my earlier pathogen, I know it. So first the
First, the
macrophages will go and
basics, macrophages will carry on
neutrophils.
his duty.
So
Okay.
first, at the site of any kind of pathogen entry, first the innate cells only go and bind
Okay.
to it.
Okay. Okay.
Neutrophils or macrophages or whatever.
Okay.
Yeah.
Thank you. Thank you.
Okay. So I hope it was helpful. Anybody has anything more to say? And then I'll be just saying bye for today, and we'll meet again next Friday. I'll just get back to you, say, like, I'll just see whether the PAMs are associated with B cell recognition or not, and in the next class. Is that fine?
Okay, ma'am.
Yeah. Okay.
Thank you so much, ma'am.
Thank you.